So, welcome everybody. As most of you know, I'm Geoffrey Guy, the founder of the Guy Foundation, and this is the second session of the spring series on magnetic fields and biology. And I'm very much looking forward to our talks today from Rhys Mould, Margit Egg, and Martyn A Sharpe. I'm going to invite Rhys Mould, who's head of group at the Guy Foundation Quantum Biology and Bioenergy Laboratory at the University of Westminster in London, to introduce the topic and also introduce our speakers today.
Good morning or good afternoon, everyone. My name is Rhys Mould. I'm leading the Guy Foundation Quantum Biology and Bioenergetics Lab at the University of Westminster, and it's my pleasure to introduce the second in our spring series on the therapeutic use of magnetic fields. So last week, Johnny Woodward and Brian Ross gave us a fascinating intro into how magnetic fields can interact with our biochemistry and biology. And we returned back to a favorite in quantum biology, the model by which birds or migratory birds can navigate using the Earth's magnetic field. And to recap really quickly, molecules called cryptochrome in the bird's eye can be excited by blue light, at which point they form this radical pair. Let me just switch on my pointer. These radical pairs, which can oscillate between the singlet and triplet forms, depending on the orientation of the spin of their unpaired electrons. Let's say, hypothetically, the singlet form of the radical pair can go on to downstream chemistry, which produces a signal that can be interpreted by the bird's brain. Now, the clever part is whether you find the radical pair in the singlet or the triplet form depends on the bird's orientation relative to the Earth's magnetic field. So, for example, if the bird is orientated, you get hypothetically more of the singlet form of the radical pair, which goes on to form a product, which goes on to form a downstream signal, and thus the bird can detect the magnetic field and use it to navigate. So the key concept in all of this is spin, the fundamental property of subatomic particles, and spin is something that's going to come up again and again in this series. And remember, it's not literally the spin of the subatomic particle. But it does, however, generate a tiny magnet, and these can be sensitive to external magnetic fields. And this is the mechanism or the way that we are presented with a grapple hook in which we can maybe be able to influence biochemistry using external fields. And so the question we ask today is, how can we utilize this feature therapeutically? So if you think about ways in which we use magnets in medicine, you might first think, because of the name, of magnetic resonance imaging, MRI. Now, instead of the Earth's measly 56 microtesla field, we use a strong field, 1.5 Tesla and above, which is called B naught. The principle and the concept are quite similar because we're still utilizing spin. Instead of electron spin, now we're using proton spin found in your body water. Outside of B naught, you have a random orientation of your protons, but then when you turn B naught on, you end up orientating the protons up or down, mostly, let's say mostly up, and they become ordered relative to the external field. Now we use a radio frequency wave to knock them out of orientation. And then when that pulse turns off, they all flip back to their original conformations, and in doing so, release energy, which we can interpret as a signal. And because this energy is dependent on the environment in which the proton is surrounded, we can use that to generate very high resolution images of soft tissue in the body, and it is the gold standard of medical imaging. As an interesting anecdote, side note, patients in even higher strength fields, such as 7 Tesla, I think they've even gone up to 11 Tesla, report some interesting symptoms, including vertigo, nausea, and metallic taste. So further evidence that we can manipulate physiology using, admittedly, very strong magnetic fields. On the other side of the spectrum, you have the technique of magnetoencephalography, or MEG, in which we detect magnetic fields generated by neural electronic activity. And of interest is when this neural activity is perturbed, such as in epilepsy. You can see the photo is a guy sitting within the detector, and the images on the right are areas of perturbed magnetic activity superimposed over the MRI of someone's brain. Now, because the magnetic fields are so very tiny, we are required to totally eliminate any background magnetic fields, including that of the Earth's geomagnetic field, the GMF. And to do so, we use something called mu metal, and rheum's made of a material called mu metal, and that is something I'm going to come back to in a bit. So these really are magnetic fields as tools in medicine, and what we're interested really in is magnetic fields as therapy. So to sum up, the body both generates and is sensitive to magnetic fields, and the question is, can we leverage this? As a side and as a note, I think it's important to note that weak fields generally are reported to have negligible effects on health, and the mean exposure of a human to magnetic fields is also very low. So devices such as your phone, telephone wires, your TV, your hairdryer, which generate magnetic fields, all do so, but they do so below what's the recommended limit for magnetic field exposure. So one of the questions we also have, and this is a kind of an aside, is what about no magnetic field? So life has evolved under the presence of a global geomagnetic field, which is around 56 microtesla. And one of the things we've been interested in is what happens if we take away that field. And apparently it's quite a lot. So I'm using this review by Sarimov, Hypomagnetic Conditions and Their Biological Action, hypomagnetic referring to anything below the GMF. In vivo, it's been reported a plethora of effects, including increased red blood cell count, rather ominously increased number of mistakes, decreased number of offspring. In vitro, it goes on and it gets more complicated. You have differential expression of genes. You have decreased mitochondrial respiration through a decreased oxphos rate. You have differential changes in cell differentiation rate. And these are all tissue and cell dependent and field strength dependent. So it's very much an insight into how magnetic fields may be able to influence physiology, and therefore how we may be able to utilize it in potential therapy. It also gives me an opportunity to shoehorn in our research at the University of Westminster to hypomagnetic field exposure as part of the Guy Foundation Space Health Program. So here we're testing the effects of hypomagnetic field exposure in vitro using the mu metal, which I spoke about earlier. Mu metal is a nickel alloy that possesses a property called permeability, which means they can block out static or very low frequency magnetic fields. So the picture on the left is our incubator in our lab, and the mu metal chamber you can see here consists of several concentric cylinders of mu metal, and inside we estimate the field to be around 50 nanotesla and below, so an order of magnitude less than the GMF. And we use this in the context of space health. So what are the implications for cell physiology beyond the Earth's orbit? So as astronauts and space travel ventures further from the Earth, what's going to happen to our astronauts on top of the effects, including such as hypergravity, altered circadian rhythms, and it's not something that seems to have been very much considered in the past. I'm going to present some very preliminary unpublished results. So this work was undertaken by Iffy before, and she looked at the recovery of a scratch in the scratch assay in C2C12 muscle cells, and we found that the rate of recovery was inhibited in cells incubated within the hypomagnetic shield. We've also been looking at the effects of hypomagnetic fields on C2C12 mitochondrial function. This plot shows the rate of oxygen consumption over time in response to a number of drugs in an assay called the seahorse, which is one of the gold standard tests for mitochondrial functions in vitro. And again, you can see that cells exposed to the hypermagnetic field for 24 hours seem to exhibit a decreased oxygen consumption rate across the whole test period. But that is enough of my research, and it's my pleasure to introduce today's main speakers. So first, we have Dr. Margit Egg from the University of Innsbruck. Margit's research focuses on the interactions between circadian clock and hypoxia signaling, and as well as how low-level electromagnetic fields can influence both pathways. And her talk today is tNMR as a resonant modality within therapeutic weak magnetic fields. After Margit, we have Professor Martyn A Sharpe from Houston Methodist Hospital. Martyn has a background in biochemistry and bioenergetics, and his research is centered on the proliferation and death mechanisms of neuronal cells, particularly in the context of developing treatments and therapies for glioblastoma. And today his talk is going to be titled Rotating Magnetic Fields and Cancer. I think we mentioned Cameron, our third speaker, was unable to attend today, so it's just Margit and Martyn. And with that, thanks for listening to my intro, and I'm going to hand over to Margit and Martyn for what promises to be some really fascinating talks. Thank you so much.
So thank you, Rhys, for your kind introduction and also for doing a recap on nuclear magnetic resonance, which helps a lot, since the device I'm talking about today is the therapeutic form of nuclear magnetic resonance, which is way weaker. And I will show you that it is a very special modality within the weak magnetic fields, although it is still in the range of weak magnetic fields. And for this, I first want to show you the setup of the device itself, which you can see here. This is an old-fashioned device we use for our experiments, and it was used for the treatment of knees by the company. And the device itself was directly deduced from nuclear magnetic resonance imaging around 25 years ago. But as I already said, in a way weaker intensity. And as you see on the left side here, it consists of 3 coils, which are located here, here, and here. So there is a sweep field, a so-called sweep field in a parallel configuration, which produces a field intensity of 400 microtesla and a frequency of below 10 hertz, which is basically close, very close to static magnetic field. And then you see here at the bottom in the socket of the device, there's a 3rd coil, which produces the radio frequency. And the radio frequency itself has an even weaker field of below 100 microtesla and a frequency of up to 17 kilohertz, which is the Lambda frequency of water protons under the conditions of 400 microtesla. So, and this is very important also, that the 3rd coil in the socket is perpendicular to the other ones. So for us, the device was a total black box over more than 10 years. Let's put it like this, like a blue box more. But 1 year ago, we made an agreement with the company because we got so many critiques about that it is like a box in the range of weak magnetic fields. So why talk about water proton resonances or nuclear magnetic resonance when the cellular effects we see might only be simple weak magnetic field effects? So we split the fields into the sweep field using controlling the upper 2 parallel coils and compared the cellular effects to the radio frequency coil in the bottom and to the whole tNMR configuration. And the manuscript is currently under review, but I will show you 2 slides and at the end, because I think this is most important to be able to see what it that it's not simple within the weak magnetic field effects. And so this is a small spoiler alert. It does something very specific, but let's move on. So as I already said, patients are still treated with the device, and in conditions like osteoarthritis, osteoporosis, or wound healing. And they are treated like 1 hour per day, and they make different cycles between 7 up to 11 sessions, depending on the severeness of the disease, on consecutive days. So this is quite difficult to make with cell culture. There's among you who are familiar with cell culture know that after 5-6 days, there's so much waste product that you have start right from the beginning, because cells overgrow and there's lactate accumulation. So we did a phase response, a dose response curve, maybe around 10 years ago also. And we used a single 6-hour exposure, because there we get most of the effects. And additionally, we used 1% of hypoxia during the treatment. And we did this on purpose, because since the devices are used for humans for the treatment of bone or joints, there you have very low oxygen tensions, down to 1%, as I said, because there's no capillarization. So this is a very natural physoxia. However, when we put them in the device under hypoxia, it is pretty clear that afterwards reoxygenation occurs. So our experimental model, which we used for all of the papers, was a simulation of hypoxia reoxygenation model, which is close to the ischemia reperfusion injury model. And for those not so much familiar to this medical condition, this occurs in clinical context. Here below, you can see it like infarct, stroke, or acute kidney injury, or also wound healing. And this means that when you have, for example, a myocardial infarction, the blood flow will be blocked. And this is not good, but most of the damage will occur when you, after some time, have the medical intervention and the blood flow is re-established, and so is the oxygen supply. And with the oxygen supply, then you have a huge burst in reactive oxygen species. And this is sometimes even more difficult to handle than the infarct alone. So please keep in mind, we from the experimental setup, it's pretty clear we are here with a very special one, because we always have reoxygenation. Okay, so let's come to the first major results. Since I'm a chronobiologist, I was interested in the circadian clock of the cells, because it's obvious we all have tiny molecular cells in each cell of clocks in each cell of our body. And one of the major key player is cryptochrome. And of course, we always watch what's going on in bird magnetoreception. And so our former hypothesis already 10 or even 15 years ago was maybe weak magnetic fields do not affect only the cryptochrome in the bird's eye retina, but also in all of the cells. And this is why we then finally, around 2 years ago, we took a reporter cell line, a period 2 reporter cell line. It's a mouse cell line where the promoter of the period 2 gene is linked to a luciferase, so that you can see the circadian oscillations of the living cells. And this is what you can see here in the upper graph in black and also in the lower one. And circadian means one peak during 24 hours. And you see that this is really nice, a nice oscillation here, first, second, third peak, then it starts to slow down. And the reporter system we used was also not only because period 2 is one of the key clock genes for the circadian clock, but it has also a huge impact in immunity. So the circadian clock plays a huge role. And so our trial was first, you see in the upper graph, when we use hypoxia only for 6 hours and then start the recording. And this was done as a control experiment for us because it is well known in the chronobiological community that hypoxia alone dampens the amplitude. As you can see here, dampens means the amplitude goes down a little bit, and you see also a phase shift. And this has been reported several times for all the mammalian systems, for all the cells and tissues. And in comparison, you see when we use 6 hours of therapeutic nuclear magnetic resonance without hypoxia this time, and you see we have an increase in the amplitude, a huge increase. We can't call it still a real Zeitgeber, which is, for example, light, because we did no phase response curve, but we did something else. You can see here that there's a sun, and this sun means that we treated the cells during the morning hours between 8 in the morning and 2 in the afternoon. And then we repeated the same treatments during the first hours of the night, 8 p.m. to 2 a.m. And you see they react totally different compared to the day treatments, but in a very similar way. So if you use hypoxia, it seems quite similar to the tNMR device itself. You see a strong dampening of the amplitude, but also the whole promoter activity drops dramatically. And from the chronobiological point of view, you could say, okay, it halts, it stops the clock somehow. This seems like something magic. Maybe if we hadn't done it with hypoxia only, we would have thought, what an amazing device. But the reaction lies more on the side of the cells, because all cells, also cancer cells, do have a circadian rhythm in the redox state, in the overall redox state of the cells, which means a cell can be either overall more in an oxidized state or in a more reduced state. And this is oscillating during day and nighttime. And this is true also, as I said, for cancer cells. So I think with the device, you really are able to address the circadian clock of cells and tissues. But most of all, we think it's more important for the treatment with weak magnetic fields, for example, because timing then matters a lot, because you get a totally different readout in the cell, a totally different response. And also from the pathological side, the circadian clock plays a huge role in stroke, infarct, wound healing, and also in oncology. Let's focus maybe on infarct. It is well known that infarcts themselves, or all cardiovascular events, do have a strong circadian rhythm. They mostly peak during the early morning hours, because there's most pressure and work on the heart, or in the evening hours. And also the response, how well you respond to recovery, depends on the time of day. So the circadian clock matters a lot in health and disease. And to see that there is a possibility to address this, to work like a real Zeitgeber is amazing. And only to show you that this was not only the promoter activity affected, we saw huge changes also in the transcription levels of major clock genes like cryptochrome 1, clock 1, and period 1. And I totally forgot to say, we use NIH 3T3 mouse cell models. So the translation to human cells still has to be done, but we are on the way also on that. And you see here basically in blue that the copy numbers are somehow reduced or also changed in their pattern after tNMR irradiation, which means that also mRNA and protein levels are affected. I didn't show too much because I wanted to keep the time for the talk. So then in 2020, we were able to publish a study because we have a lot of knowledge in Innsbruck regarding the circadian clock and its tight interaction with the hypoxic signaling pathway. And the hypoxic signaling pathway is under control of hypoxia-inducible HIF-1 alpha, which is a transcription factor which governs and controls cellular glycolysis and also the branching point between glycolysis and mitochondrial respiration, because it depends on the oxygen amount available for the cells. So please keep in mind here the data we show you. Rhys mentioned already, it's the Seahorse flux analysis we did, and on the left side you can see the oxygen consumption rate. And please compare first the black ones, cells, living cells after 6 hours of normal oxygen supply. And then in red, you see cells which experienced hypoxia of 1% for 6 hours. And for those not so familiar what this could mean, I compare it always like if I press you underwater, and you have a lack of oxygen, and then you resurface, you will breathe deeply because you do have a lack of oxygen. And so this basal respiration is heightened after hypoxia. And when we add tNMR to the hypoxic conditions, this does not happen anymore, which is quite strange. It's closer to the control cells than to hypoxic one. And on the left side, you see the ECAR, which is basically the extracellular acidification rate, which gives us an estimation of CO2 and lactate production leading to acidification then in the cell. And you see lactate and probably CO2 increase after hypoxia, which is quite common. And again, when you add tNMR, it is lower. So in 2022, we were totally happy to see this. And then we tried to reproduce it all together later on. And it turned out to be the other way around. And we found out also why, because in this setup, we used dexamethasone, a corticosteroid, to synchronize the circadian clock of the cells, because it was our topic for the manuscript. But when you don't use dexamethasone, the readout is very diverse. So it can go also into the other direction. And I will tell you later what this basically means. And still in the field of glycolysis and mitochondrial respiration, you see here we did a totally different measurements. We used the metabolite section of dead cells after the experiment. And we measured many. I only showed you 3 of them because otherwise it's confusing to see, like intracellular glucose, pyruvate, and intracellular lactate. And here it is important to mention that those are steady state levels. So we can't really tell you if it's more used or less introduced into the cell. We can't tell this, but we could have a basic idea on what happens. And so the trial was the same as in the slide before. And you see under hypoxic conditions, glucose is depleted compared to control cells as it is for pyruvate. And we see the typical increase in lactate, which happens also when you go high up in the mountains where oxygen tensions drop. And you also see from far away that obviously when you add tNMR, it makes a difference. Especially for lactate production, which we saw that it was decreased as well as we saw in the data from the seahorse flux analyzer. Though there were differences because glucose remained in between and pyruvate was significantly lower. Okay, and then the third very important point, which we did, most of all because of the interest in the quantum biological field, because the research on the radical pair mechanism and those differences. And that's why we wanted to measure reactive oxygen species, but compartment specific with different methods, which should be quite up to date, I think. So you see here in the upper graph, the measurement of mitochondrial superoxide, which we did. On the right side, you can see it with a laser scanning microscope using MitoSox green. And we did an H2O2 measurement in mitochondria using a raw GFP or one targeted mitochondrially targeted cell line. So it's a reporter cell line, and measured between 0 and 6 hours after the treatment, which means, again, reoxygenation, everything. And we were quite puzzled because biologists like when it's traditional, like it's always been in the textbooks. And for us biologists, normally superoxide is the first one showing up. And if levels get toxic, so the anti-defense mechanism will show up. And in this case, the superoxide dismutase will transform superoxide into H2O2. And when those H2O2 accumulates and gets toxic in the cell, catalase breaks it down to water and O2. So normally we have first superoxide and then H2O2, but this was not the case here. It was chronologically not linked. So we got higher amounts in the end of superoxide and initially increasing amounts of H2O2. But what was even more puzzling for us, and this happened only because Vicky was so curious and she wanted to see if the whole situation could change if we use MitoSox during the incubation with tNMR and hypoxia. And so we did this trial as well. And the outcome was really a little bit disturbing because all of the cells died. You see it here. They got a very spherical shape. They detached from the Petri dishes and they died. You see here, 80% of the cells died when we used MitoSox together with tNMR. So our thoughts were, maybe it could be that MitoSox does interfere or is a form of stress for the cells, or it's because the dye, this fluorescence dye, works as a scavenger. you will get the fluorescence signal when superoxide is scavenged by the cells. And that's why we made the same trial once again, hypoxia in combination with tNMR, using the scavenger NAC or vitamin C. And then the right side, you see the same thing happen. And until now, we are not quite sure why this happens. We want to do further experiments on that. But we know that the major problem were the increasing amount of H2O2, which then result in higher concentrations of the hydroxyl radical via the Fenton reaction. And this happens also with cancer cells. Okay, so those are the major findings. We did a lot more work, but it would be too confusing. So let's sum this up before going on. So what we can tell, and I'm quite sure that this might be true also for any application of weak electromagnetic fields, the circadian redox state determines the responsiveness. So this is very important, and this owns also a huge potential if we think of doing night treatment, for example. Then what we see from all our studies, not only the slides I showed you, but all the studies we did on carbon flux, lactate handling, oxygen consumption, we saw that tNMR always induces a distinct redistribution of glucose, lactate handling, or the branching point between mitochondrial respiration and glycolysis. And so I already mentioned the oxygen consumption rate also, it changed significantly, but was heavily influenced by the context, which means in this case, plus dexamethasone, totally different than without. So it's not like a black and white that you can say, oh, tNMR increases the oxygen consumption rate. And then we do get a very specific Ross signature, which changes as well, context dependently, and it's compartment specific. So we see we measured also, for example, H2O2 in the cytosol, and it changes as well after tNMR treatment. And because of the slide I showed you before, because of the cells dying when you use a scavenger during the treatment, we interpret as that superoxide has to have a very essential function in the downstream signaling response to weak magnetic field irradiation. So we are quite sure that this is a very, a stable interpretation of what's going on. So this means that we have already a lot of therapeutic implications also. So our setting here, we are very clear, is a model for hypoxia reoxygenation settings. We can't go further. It's not under chronic hypoxia. And we can say that timing and the baseline redox state shapes the responsiveness. And we're quite sure that this is true for human cells as well as it is for cancer cells, because we did already reviews on that. And this is the case. And then we can tell that obviously mitochondria seem to be a primary response node. And I think this has been found by several groups already. And I'm sorry, I forget to mention the story with the superoxide as a central signal mediator. This was also found by the group of Wendy Bean. She did really cool experiments on that. So also from different groups, very similar findings arise. And then finally, OCR and lactate are context-dependent outputs. So one has to consider them more globally than interpret too much in a special situation. Okay, so initially I gave you a spoiler alert, but for us it was not only for us. I think it is important for the company, but also for the field itself, for the weak magnetic field effects, because we were able to split the fields and did some trials using only the parallel configuration, which I think would be most similar to the weak magnetic field effect experiments going on, using maybe different field strength, but no high radio frequency. And then we have the radio frequency call, which is a little bit odd because, as I told you, the radio frequency is 17 kilohertz. So from the physical perspective, it should not affect electron spins, regarding now the question if the radical pair mechanism could be involved. And then we have the whole configuration. And what we found out by really measuring a large amount of varying parameters in the cell, we got totally different states. So for the weak field, we got the redox shifted state, which is characterized mostly by real extracellular H2O2. I think this is really in line with what we know how weak magnetic fields affect cells. And we saw that mitochondrial H2O2 decreased. And from the seahorse flux analyzer, we know that the spare respiratory capacity increased, which is a good sign. And we gave all those measurements that, as I said, we made many, many more of them and run it through a deep search in AI. It's mainly repair and inflammation, where this setup should be very helpful. And then let's go to the right side, DRF conditions. So we have high intracellular glucose and pyruvate, whereas lactate and ATP go down. And the most interesting story is that the spare respiratory capacity goes down, so that the phenotype of the cellular state is clearly carbon biased. But regarding the respiratory capacity, it is restrained. So AI would put this in an interesting option for oncology itself, because the cells obviously are stressed when you use the radio frequency alone. And we have a quite low field intensity, as I mentioned before, around 100 microtesla. So, and for tNMR, which, so for tNMR in all the cases, be it with dexamethasone, without dexamethasone, in all our publications, we saw an increase in mitochondrial superoxide, and not a small one. And we always saw an increase in ADP plus and ADPH, whereas the OCR and the lactate handling was clearly context dependent. And since lactate itself and mitochondrial superoxide and this ratio here, we tried to interpret that what might be the underlying biological mechanism, and we think that it could hint at a proton transfer linked like state, which means that proton kinetics are affected. And for the therapeutic use, chronotherapy is an option, but obviously regeneration and also ischemia reperfusion. And indeed, one hospital in Germany already makes some trials to treat people facing a blocked blood supply after infarct or a stroke, and treating the tissue in between the infarct and until the time point when the medical intervention comes. We don't have any data on that. But what I wanted to show you is that those arrows, those tiny arrows, are not only trends, which we saw when we split up the fields, but really huge differences, as you can see here. And this graph, you see the NADP plus NADPH ratio, and you see normoxic cells, no, sorry, I'm wrong here. You see the RF field, the sweep field here, control cells with no treatment, and here tNMR. So it is heavily increased. And this is true also for mitochondrial superoxide. So levels differ a lot, and we found also major differences in the RF field regarding, for example, the reduced spare respiratory capacity I mentioned. Okay, so I gave you an outlook what's going on in Innsbruck, and this manuscript is currently under review. And of course, we hope that it can give some inspiration or also for the field itself, because we strongly believe that those proton resonances could be helpful also for further study of the radical pair mechanism itself. And of course, we try to connect with quantum physicists on that point. Okay, and finally, I want to present you the team in Innsbruck. Fortunately, Victoria is still working with me as a postdoc because this is a lot of fun. And Cordula is here, and also Justin. They're our master students, and we can keep them if they want as PhD students. So work is going on. And here you see Vicky in Finland with the Finnish group, Thomas Kietzmann, who is a ROS specialist also in cancer, and Eliza Dimova, both from the University of Oulu. She's a chronobiologist like me. And Hadi Zadeh Hagigi, whom you might know, a quantum physicist working in the field from Canada, Calgary. Okay, thank you very much for your attention.
I'm Martyn A Sharpe, and I'm at the Kenneth Peak Center for Brain and Pituitary Cancer Treatment and Research at Houston Methodist Hospital. This is my confidentiality thing. I got to tell you, I got a dog in this fight, so you don't necessarily have to believe everything that I say. So we've got a patent with Santosh Halaka, who's a biophysicist, David Baskin, who's an actual brain surgeon and treats real patients, and Kumar Pichimani, who's an expert in NMR and MRI. We have a financial interest in oncomagnetics, and this company is developing this treatment, and we were given some money by a group called CPRIT, which is part of the state of Texas, to bring this technology into clinical trial. Okay, so Nina begged me to give you all the mathematics behind statistical mechanics to explain what's going on. She said she wanted a real in-depth analysis of quantum theory. The problem is that if you study statistical mechanics like Boltzmann or Ehrenfest, you tend to die by suicide. So I will give you a more simplified overview. So everybody keeps talking about spin, okay? So for electron spin, all you've got to understand, it's like a ball that's rotating, except it's not a ball and it doesn't rotate. Okay, so the quantum world is completely different from how you perceive the world. But there's one thing that you do understand, which is a clock face. So the hands on the clock go round in a clockwise direction. But if you looked at the clock face from the other side, it would be going counterclockwise. So spin, the property of spin depends on the object and your orientation. So the what we call spin is basically the same as an object spinning round, and it has got either one direction or another direction, clockwise or counterclockwise, and the poles give you a north-south or a south-north directionality. So what we want to know is, does spin exist in the real three-dimensional macro world? And this was answered back in 1922 by two German Jewish scientists who were recovering from the First World War, who didn't believe all of this quantum nonsense and came up with an experiment to test it. And they heated up a little piece of silver, and silver has got an atomic weight of 47, which means it's got an electron in its orbital field that is a solo. They puffed a cloud of single atoms of silver through a magnetic field and managed to get a pair of lips. And that tells you that the spin state of those silver atoms is responding to an actual field, spin up or spin down, in three-dimensional space. Okay, so spin is not only real, but it's real in our world. Oh. So where do you see spin in biological complexes? Okay, so this is succinate dehydrogenase. Okay, it has a 2 into 1 into 1 into 2 electron flux. Here's succinate. Here's the flavin. The flavin takes 2 electrons and gives electrons one at a time to 3 iron sulfur centers, center 1, center 2, center 3, and also to the iron atom of this heme. Now, when you have a flavin radical, you've got a spin. You've got an unpaired electron. Okay? Center S1 only has an unpaired electron when it's reduced. Center S2 only has an unpaired electron when it's reduced. Center 3 only has an unpaired electron when it's oxidized. And ubiquinone is the 1 electron addition to ubiquinone. Okay? So it's got 6 radical centers. So you can actually oxidize 3 succinates at the surface of succinate dehydrogenase. And like in baseball, you can load all of these centers with electrons without ubiquinone being present. Okay, and that's one of the causes of reperfusion injury. Okay, so what's a biradical pair? We've got 2 centers right next to each other, like 2 iron sulfur centers. We've got an oxidized electron acceptor and nothing on this side. And we're going to reduce the donor. Now, the spin of the donor can come in with the same direction as the electron acceptor or in the inverse direction. Okay, actually any orientation. But this state is called singlet, and this state is called triplet. And it makes an awful lot of difference to the sort of chemistry you see. Now, biologically, we favor putting the electron into here so that it spontaneously forms a singlet state, because this is what's known as spin favored. And the reaction is very fast. The electron forms a quantum state, the pair of electrons form a quantum state and suddenly become paired. Okay, and you've just had electron flow, and that's all pretty normal. The other way we can do is we can make what's called the triplet state, where both spins are in the same direction. And this guy cannot move to this center because it's spin forbidden. Both electrons are in the same state and they can't occupy the same state place in the same state. Okay, so we do not get to here. And if we generate a triplet system, what generally happens is we get a reaction with oxygen and we form superoxide. And superoxide is very nasty, and we get rid of it as quickly as we can by turning it into hydrogen peroxide, which is considered less worse. Okay, so these are reactive oxygen species. Now, magnetic fields are also real and are in three-dimensional space. So this is a little bar magnet, and what you do is you distribute iron filings and you can see the lines of force. And the lines of force are essentially spherical. Okay? So this is the manifestation into the macro world of actual quantum events. So if you took a line in this direction, it would be telling you that you've got peaks and troughs of magnetic field strength. But these fields are changing orientation as you go from here to here. So I bought 20 little magnets, compasses from Amazon, and 19 of the 20 pointed north-south, and one of them pointed south-north. So if you're going to take a bunch of school kids on a hiking expedition, don't buy your compasses from Amazon. Okay? Here is one of the magnets that we use in the treatment of our patients, and it's in the north-south orientation. And you can see that this magnet is north-south and this one is north-south. They're in the same orientation. But as you do 90 degrees, you see that this guy is flipped 180 degrees. Okay? At 45 degrees, it's on this side, I can't see it, but at 45 degrees, you get a horizontal flip. So every time you rotate this magnet once, these guys will rotate twice. Okay? So these exist in the macro world and are responding to this magnetic field. What's that got to do with cancer? Come on. Okay. So we're back to our little pair of centers, and this oxidized one has got a single spin, and we've got a rotating magnet. Okay, it's the rotating part that's important to understand. When the field is aligned with this spin, it essentially becomes locked. Okay, in the same way the little compasses were locked. And as we rotate, it follows the rotation pattern. And it's no longer in the biological direction that evolution has prepared it for. And we put a second electron in, we put an electron into the reduction state, and we get both of those electrons in the same state. Okay, so not only is there no reaction, not only can this guy not go to where its normal destination would be, but we've trapped it in a triplet state, and in that state it can react with oxygen, form superoxide, hydrogen peroxide, Fenton chemistry, all the guys in the band, all the way down to apoptotic cell death. Okay, now that's a big claim. Okay, so succinate dehydrogenase, again, a 2 into 1 into 1 into 1 into 2. Now, there are 3 states that we can put to form triplets. Okay, we can have the flavin semiquinone and the reduced center 1. That would stop the back reaction. Okay, we've got the final state, which is the oxidized center 3 and the UB semiquinone, and we can block the back reaction, but not the forward reaction. And then there's this guy. So we've got the reduced center 2, oxidized center 3. They are both radicals, and we can hold up the electron here. And if we hold up the electron here, what we should do is stop flux through superoxide dismutase on to making quinol and stop mitochondrial respiration on succinate, and also generate superoxide, hydrogen peroxide, all the guys in the band. So how do we show this? Okay, So this is an oscillator. This is one of my actual oscillator magnets. This is one of the research magnets that we use. And all this is a high-speed motor with a little magnet stuck on the shaft. And we spin it up to 280 hertz. And then we decelerate down and accelerate it up and decelerate down. And I'm not telling you how much we do that for, right? Because it's super secret. But what it means is that we're doing a frequency scan at the same time that we're rotating the field. And we stick the magnet on the end of a microphone stand, and we put the microphone stand next to an oxygen electrode. So this is a little picture of my oxygen electrode. At the base of the oxygen electrode, in the Z frame, we've got a rotating magnet that's got a stirrer bar on it, because you need a stirrer bar above your platinum membrane. And here's our oxygen sensor. Here's the fluid where we have our mitochondria or our cells. And alongside, in this orientation, we've got the oscillator. And it's air gapped, okay? There is no connection between the mitochondria and the magnet. We got a magnet, we got air, we've got the water jacket, and then we've got our mitochondria. So I love rat liver mitochondria. I can play with them for hours. So here's an oxygen electrode trace. We add our rat liver mitochondria. They're not very energetic. We add succinate, and they start consuming succinate. They love it. And then we switch on the oscillator for 5 minutes. And we get a complete cessation of oxygen consumption. And then we switch it off. And it goes like the clappers. And then starts slowing down. That is where the mitochondrial permeability transition has happened. What we did in this 5 minutes is we collapsed mitochondrial membrane potential, and we were also producing superoxide, hydrogen peroxide, general Ross species. And we know this because we can block this with cyclosporine, we can block this with bemcrickic acid. When we switch it on again, we also go flat, okay? And that's a clue. We switch it off, we get our rate back. So we do the same experiment, but this time we do it in the presence of an uncoupler, so there's no mitochondrial membrane. Now, if you give succinate to mitochondria that are uncoupled, they go really, really quickly. So what we wanted to do was give a competitive inhibitor called malonate, and that means that we have got the same turnover here as we had here, okay? Because we want to see what the turnover is like in normal physiological states. So this is our control. We add our rat liver mitochondria, we add our succinate, and you can see that we're linear for around about 60% of the trace, which is normal. And then we do the experiment with the magnet, and we switch the magnet on, and the respiration drops to 66%. Second cycle, 45%. Third is 33%. Then we had beta hydroxybutyrate, which is our control. Here, electrons enter the mitochondria through complex 1, not through succinate dehydrogenase, and the mitochondria are still functional. We can repeat the experiment in the presence of Trollocs, which is an artificial version of vitamin E. It's a beautiful antioxidant that works in aqua and in membranes, and you can see that we got no effect. Okay. So the rest of what I'm going to be talking about is the effect of oncomagnetic stimulation on tumor cells or tumor growing in a patient's brain. And the tumor is called diffuse intrinsic pontine glioma until around about 18 months ago, when it was reclassified as DMG for diffuse midline glioma. It's the same tumor, and it's horrible. This normally affects children. About 15% of the cases tend to be young adults, and the overall survival, median survival, is somewhere between 9 months and 12 months. So we're using clonogenic assays and isolated cells with inhibitors and activators to do a forensic analysis of what's going on. Okay, so what we do is we have a sham treatment, and our sham treatment is normalized to 1 in terms of our survival assay. So we put dilute cells on a plate and wait 3 weeks. We dye the cells and we count the number of colonies. So each colony comes from a single cell. If we do 4 hours of treatment with our oncooscillator, we lose around about 40% of the colonies. Okay, so this is the kill rate, 40% in 4 hours. What we can do is we can activate the NRF2 antioxidant response, okay, with a drug. And here's the drug on its own. And here's the drug with magnet. And calc shows you this fraction multiplied by this guy, and it tells you what you expected to see was this. And you can see the red one is very much smaller than this one. And that's telling you that if you activate NRF2, you get complete protection from oncooscillators. Okay, so that tells you it's reactive oxygen species. And you do the positive control, and what you do is you inhibit NRF2 responses, and it's essentially 100% lethal. Okay, so ROS and antioxidant responses. So how do we know it's mitochondria? Well, we do cyclosporin A, which stops the mitochondrial permeability transition, and we get complete protection. We can add an inhibitor of caspase 3. Okay, we add a synthetic peptide of caspase 3, and again, we get close to complete inhibition of cell death. Ferrostatin 1 was suggested by a kind reviewer, okay, who was interested in ferrotoposis, and it does you absolutely no good at all. This isn't iron release generating some strange pathway that some people believe in. Now, the amino acid serine is the substrate for the 1-carbon pathway. The 1-carbon pathway can operate inside mitochondria, and there's also in the cytosol a 1-carbon pathway. And both these pathways produce NADPH, but it's compartmentalized. NADPH does not move inside and outside the mitochondria. So you add serine and you afford complete protection from SOMF. Okay, so one of those 1-carbon pathways is producing NADPH and keeping the cells alive. So what you do is you inhibit the one inside the mitochondria using a specific inhibitor of the mitochondrial 1-carbon pathway, and you see that you get more cell death than you expected. Then you use an inhibitor of the cytosolic pentose phosphate pathway, which is the main way that cells generate cytosolic NADPH, and it really doesn't potentiate SOMF. So we know that ROS is what's the killer, and we know that it's mitochondrial, and we know that mitochondrial NADPH is the thing that rescues you. So this is what happens when we had a broad spectrum antioxidant, Trollocs. Trollocs is completely protective. Okay, now Trollocs is very nice in that it protects membranes and cytosols from the effects of reactive oxygen species. We can do the time dependency, and you can see that the longer you stimulate the cells with SOMF, the more toxic it is. And as an internal control, we use radiation. So 1 gray of radiation is approximately 4 hours. Now, in normal brain tumors, we radiate the patient with 2 gray once a day for 6 weeks. With our patients, we give them 2 sets of 4 hours a day, forever. Okay, so I don't like the seahorse system because we lose cells, and I just spent $170,000 on the Respire system, which is an optoid system that allows you to measure oxygen consumption rates in 96 well plates. I got a pair of them, and what we'll be able to do is do sham treatment and SOMF simultaneously and measure the oxygen consumption over the course of days. This is the preliminary experiment that I did with the loaner device, where we used mu metal to shield half the plate. And what you see in our sham controls is this exponential rise in oxygen consumption, which is due to cells doubling. Okay, so we know from looking at this trace that what we've got is a turnover of 20.4 hours. That's our doubling time. And then at this point, we added oligomycin on this trace. In some of the traces, we added DMSO as our vehicle, or FCCP. And from that, what we can do is we can reconstruct a Martin brand seahorse style experiment, and it shows us what the reserve capacity is, how much ATP these cells could produce from their mitochondria, how much they're actually producing, and the leak. Okay, if we do two treatments with SOMF, what you see is that we've got mitochondrial damage. The actual level of maximal capacity is down, and the proportionate leak rate is higher. So these cells, these mitochondria, are what's technically known as screwed. Okay, we have damaged them and they're leakier. So the amount of ATP these cells can generate from their mitochondria is down, which means that if you give them any stress on top of this, you'll kill them. Okay. Here is a sham, and here is an SOMF treated. And what we did in a flatbed fluorimeter is we loaded the cells with a with a peroxide reporter under 3 conditions. We had DMSO, we added an inhibitor of the pentose phosphate pathway, and we added an inhibitor of mitochondrial NADPH production. And we just let it run, and every 12 minutes we ejected the plate, and here we sat the plate over the oncosylator that wasn't spinning, and then we shut the door and we did it again and again and again. So we've got to be very careful using strong rotating magnetic fields with electronic equipment, because they don't pair very nicely, because they produce eddy currents and we can do a lot of damage. Okay, but what this takeaway is that if you inhibit mitochondrial NADPH production, you increase the rate of peroxide production, and that's more than you get if you inhibit the pentose phosphate pathway. And that's essentially telling you that most of the hydrogen peroxide you're producing in these cells comes from the mitochondrial matrix, which is something that's basically true. Here we do the same experiment, but we rotate the magnet, and you can see that there's a jump. Each time we have the magnet on, there is a jump, and that jump is around about 2.4 times the background level. So when we're running the magnet, we're guessing that what we're doing is we're producing around about 2.5 times more hydrogen peroxide. And you can see that we've got an increase in the amount of hydrogen peroxide. The rate of hydrogen peroxide is higher in the presence of these 2 inhibitors. We also use mitoSOX, and we measured the steady state level. Every time we turn the magnet on, the amount of mitosocs, the rate of mitosocs generation increases. So we switch it on, off, on, off, on, off. And we did this in the presence, in red, of the inhibitor of mitochondrial NADPH synthesis, because some people demand that you do a peroxide control, because other oxidants other than superoxide can give you a mitosoc signal. But if you look at the overlay, what you see is when the magnet is on, we've got 2.5 times more superoxide being generated at those time points in both conditions, whether we add DMSO vehicle or mitosocs. So there's another little trick that we can do, is that we can use hyperpolarizing substrates. Now, you can make mitochondria produce a hell of a lot of NADH and acetyl-CoA and succinate by overfeeding them. And the way that you overfeed them is that you give them glutamate or lactate or both together. And what you do is you measure the amount of mito tracker that you've got, and you add FCCP, and FCCP tells you when you've got depolarization, and you rank it against your control. And you can see that if you add glutamate, lactate, or both of them together, you get membrane hyperpolarization. The mitochondrial membrane potential is artificially high, or you can add serine. Okay, serine is not a hyperpolarizing substrate because it takes a while and a lot of effort to metabolize serine, but it is a substrate for mitochondrial one carbon pathway, and so it will give you more mitochondrial NADPH. So we do the same sort of experiment, loading with a detector of peroxides, and you can see in the presence of serine, the amount of hydrogen peroxide that's being generated goes down. So that tells you that what's being generated is hydrogen peroxide and not X per species, because only hydrogen peroxide is soluble across the intermembrane space. So if you generate mitochondria inside mitochondria, it can diffuse out of the mitochondria into the cytosol, but other peroxides can't. Here's our control. Here's glutamate, here's lactate, and here's lactate and glutamate. Now, each time, this jump is approximately 2.5 times your background rate. So it tells you that what you're doing when you're using your magnet is increasing the amount of hydrogen peroxide from whatever background you had previously. Okay, so it's additional to the normal level of hydrogen peroxide generation that you see in mitochondria, because mitochondria produce superoxide and hydrogen peroxide all of the time. We reckon it's around about 5% of the oxygen that your body uses comes from oxygen stealing an electron from the respiratory chain, getting converted to superoxide, and then being converted to hydrogen peroxide. Okay, so this is the first DIPG patient that we treated, and the FDA gave us permission to treat her because of the compassionate use program, because she was expected to only have between 60 and 90 days survival. So the patient wears a helmet, which has got onco-oscillators on it, and we have around about 10 to 12 centimeters penetration. But what we didn't cover is the lower neck. We didn't cover that region at first. So this is the timeline of the first patient. So January, February, March. So in April 2022, she had her first surgery. The pathology identified that she'd got a DIPG. She had chemoradiation, and she progressed 4 months, 5 months after her first surgery. And in August, it's day zero. August 2022 is when she first wore the helmet. And for the first year, we thought we'd won. Okay. And at the end of the first year, we started to see new lesions. And the bad thing was in November 23, we saw tumors in the spine. And it was in the area of the spine that the magnetic field hadn't covered because of the way that we had positioned the magnets. She had radiation therapy, and I'll show you the swelling. So we had to put a shunt into the ventricle. And then we saw improvements, and everything was getting sort of better. And I will show you the scans. But our patient, unfortunately, was feeling somewhat better, but had a fall in our home because the end-stage patients are fairly brittle. And she broke her nose and her face and ended up in the shock trauma unit. And unfortunately, it was at a different hospital to us, and they were not treating her. But she had 744 days, and she is the longest lived adult DIPG patient that we know of. Okay. So this is why we were so happy. So on day zero in the sagittal scan, you can see that she's got tumor in the brainstem, and that's not a good place to have tumor. There's no really good place to have a tumor in your brain, but this is pretty much the worst. So this is day zero, 2 weeks, 3 weeks, 2 months, 3 months. Okay. At 9 months, there is no enhancing component in any of our MRIs. If you look at the transverse section, you see this big fat guy, right? And you can actually see the center opening up, and you can see the retreat, and you can see just this little bit, and you can see it's gone. Okay, and if you look at the plots, what you can see is you can see a collapse in the enhancing area. You see the necrosis as a primary feature, and then you see the living cell volume. So we thought we were winning. Well, I thought we were winning. Okay, I'm sorry, I can't see which day that is. Okay, yeah, so this is 324. So this is the area that the field covers, and you can see this lesion here has been eaten away. So everything looked like it was doing well, but unfortunately we had a lesion in the spine, which wasn't covered by the field, which was somewhat unanticipated. Okay. Okay, day 518, you see all of these lesions. Now, you don't know whether this is tumor, whether this is immune system, or a mixture of the two. Post radiation in the neck region, and you can see that this sort of disappears. And on day 740, we see more lesions. Now, what this is telling you is that an end-stage patient has got tumor cells diffused throughout the brain that are not visible on MRI, and they're either growing, which is giving us the gadolinium labeling, or the immune system has identified them and we're getting a immune response. So we have you can use MRI in NMR mode, and what you can do is you can run the NMR spectrum of your lesions. This is our control brain, so we got a big NAA and a small choline, and creatine is slightly higher than the choline. That's what normal brain looks like. This is what this tumor looked like on day 452. Okay, so you can see NAA is down, and you've got a huge spike of choline, which is normally interpreted as being tumor. Okay. Then what we see is we see swelling, we see the appearance of necrosis in this new lesion, and we see these disordered lipids. Okay, these are a sign of tumor necrosis, but we've still got this big choline spike. You can see how large the ventricles are here compared with here, and the patient had got hydrocephalus, so we had to put a shunt into her ventricles, and they drained, and she felt a lot better. Okay, you can see this choline is down compared with the lipid, and on day 694, the choline and NAA signal are almost exactly the same as you see in control brain. So you can interpret this 150 days as treatment. You can see this little guy who gets bigger and then disappears. So what we're seeing is treatment effects on the lesions. Okay, but our problem is that we don't have all of the tools we would like to interpret the data that we see, and we are trying to move away from patients who have got very advanced disease. We want to treat them essentially as they come out of, well, before they actually go into surgery. So we've done our first full clinical trial in Germany. We got 13 patients. Yeah, we got 13 out of 12 patients, which is very Germanic. And we have different targets that we're set to meet. And I don't know, I am incredibly optimistic by what we have seen, but I can't tell you what the numbers are at the moment. But all I will say is that we have a device. This is a graphic of one of the iterations of the full clinical device. This isn't the final one, but this is the sort of thing it's going to look like. And of course, it's got a magnet closer to the nape of the neck so that we can actually treat the spinal cord, as well as the more gross features that you see in glioblastoma.